Okay, so this is the first part of Chapter 11 lecture, classifying and characterizing prokaryotes. So when you look at the study objectives, these should look very familiar, at least the first four. And the last objective will be covered in the second part of the Chapter 11 lecture. So the first four objectives, the first lecture segment here will be basically review. So of course prokaryotes are a very diverse group. They grow in pretty much every habitat. There's really only a few that are pathogens compared to the number that are non-pathogens. But since this course covers, is mainly for pre-health majors, we tend to focus on the pathogens. But keep in mind that most bacteria are actually either neutral with regard to humans or beneficial. In terms of the shapes, the ones I would still expect you to know are going to be the coccus and the bacillus. Some of you are still struggling with those terms. So those are still going to be our main shapes. We may see some that have some of these other shapes in some special cases, but coccus and bacillus still your two main shapes. Endospores. You need to remember, if you've forgotten, that they are produced by the gram-positive bacteria in the genus Bacillus and the genus Clostridium. A vegetative cell is what we call a regular cell, and in certain conditions, a vegetative cell becomes one endospore. So it's not a reproductive process, because one vegetative cell becomes one endospore. So you don't end up with two. It's not reproduction, it's just a state in which the cell can be protected against unfavorable conditions. The endospore can become a vegetative cell again later if the conditions are better. The main concern about Bacillus and Clostridium comes from people who process food, especially canned food, because the canning process involves high heat and high pressure, and that does kill most organisms, but if spores get into the food, then it will not be killed, and especially if the spores are dangerous species. And so food processors have to worry about that. Healthcare professionals, especially with Clostridium. Clostridium difficile is a secondary infection that does cause problems for patients. And of course health agencies in general. So all prokaryotes reproduce asexually. The most common type of cell division is called binary fission. There are two other types of cell division which we'll just mention briefly, but binary fission is by far the most common. And in binary fission, the chromosome replicates, and then a septum forms in which the cell membrane and the cell wall are constructed to separate the daughter cells. The cytoplasm is divided more or less equally, and the chromosome is very carefully segregated so that each daughter cell gets one chromosome. Snapping division is really just a version of binary fission, and the reason they call it that is that when the two cells separate, the old portion of the cell wall snaps to start the separation of the two daughter cells. In my mind, I don't see it as a completely different process, and only some species do this. But you still get two daughter cells, you still get replication of the chromosome, and so on. Budding, it also gives you two daughter cells, which are genetically the same, but in budding you clearly have an asymmetrical division of the cytoplasm. So an extension of the cell stretches out, the chromosome, one copy of the chromosome moves into it, and then it separates, and you clearly have a small daughter cell and a larger daughter cell. So budding's a little different, but it's you still get two, and this daughter cell will increase in size eventually to become as large as the other. So it's, again, still just another version of cell division that some bacteria do, but binary fission is by far the most common. Now, arrangements of prokaryotic cells. On this slide we see the cocci. You can have individual cells. You can have diplococci, which would be pairs. You can have streptococci, which are chains. And then the other one that's pretty common is staphylococci, which are clusters like grapes. And then these other tetrads and sarcinae are more rare. But what we'll see in lab would be individuals. We might see diplococci, which are pairs, chains, or clusters. For the bacilli, we have singles, we have diplobacilli, and then streptobacilli. Those are the three main. You can have little cluster groupings or little V-shapes, but we won't see those in lab. So single, diplobacilli, and streptobacilli will be our main three. And so that's really the end of this portion of the Chapter 11 lecture. The next segment will cover these specific examples of bacteria.